All right, so the third major chromosomal mutation that we're going to discuss is inversions. And inversions are exactly what they say. We try not to make this too complicated, but this is where you'll actually see a chromosome segment be inverted or switched around by 180 degrees. Two major types of inversions can actually take place, and we'll discuss them a little later on in this segment, but they are paracentric, meaning that the inversion does not include the centromere. And then there's pericentric, and that means that the inversion does include the centromere. So as you can imagine, those could be pretty detrimental. So to regress just a little bit, let's talk more broadly about inversions. Again, this is going to be where a segment of the chromosome is actually turned 180 degrees. So for example here, if we have our genes A and B on the short end of the chromosome, followed by our centrosome or centromere, and then genes C, D, E, F, and G, when the chromosome goes through the S phase or through the synthesis phase, and that chromosome is replicated again, and an inversion actually will occur. It could actually be an inversion that could result in gene C, F, E, D instead of genes D, E, and F. So really what's happening here is that genes D and F are switching places, but also the order of gene E is being altered from front to back. So this causes a problem because in prophase 1 of meiosis, chromosomes are going to form an inversion loop, as you can see down at the bottom of this figure, and that loop is going to allow pairing for homologous regions to actually take place. So it's kind of a messy process when we get into inversions. Again, here we see a 180 degree inversion happening, and this makes it really hard for chromosomes to pair up because they're having to find their genes that are not in the correct location. So they're forming these inversion loops. And within these inversion loops, one of the conditions of this looping effect is we can get crossing over to take place within the inversion. So this gets really messy really quickly. And as those chromosomes start to separate during anaphase, then what ends up happening is we have the two chromosomes that are connected together because the D gene paired up and crossed over in the prophase 1. And then we also get a portion of the chromosome that isn't complete and has no centromere. So as the anaphase takes place, there's nothing to connect that the microspindles or the spindle fibers to the centrosome because there's no centrosome. So that piece of the chromosome just disintegrates and is broken down and lost. So again, here we see the centromeres trying to separate during anaphase 1, stretching the dicentric chromatid across the center of the nucleus, and then the chromosome that lacks the centromere is going to be lost or acentric. So inversions can produce many phenotypic effects from an inverted gene order, and this is really important when it comes to genes that depend on their specific position to be expressed. Okay, so this is what we call position effect. And position effect can really result in some major deformities and symptoms because a lot of genes require them being in a certain order to be turned on correctly and turned off correctly. So inversions can also produce deletions as the inverted chromosomes go through meiosis as well. So if we see an inversion happen, just realize that there's a lot of different things that can happen downstream. So as we look at how this all progresses, even though we've looked at the last couple of slides on an individual basis, this is what that whole process looks like. So up here we've got our inverted loop or our inverted genes, and that results in an inversion loop where crossing over is taking place, trying to get all the genes to match up with one another. That's going to result in a dicentric bridge and a portion of the chromosome that's going to be lost because it doesn't have a centromere. And then during anaphase 1, the chromosomes are not able to separate from one another without breaking a chromosome. So that really leaves us with 4 gametes in this example. So we've got a normal gamete up here at the top, so with genes A, B above the centromere, C, D, E, F, and G. And then we've got 2 non-viable recombinant gametes that are missing portions of genes that they need to be expressed. And then we have a paracentric inversion chromosome down here at the very bottom, which means the chromosome is still intact, it's still functional, but the genes are in the wrong order. So where that chromosome can run into problems is if we have genes that are affected that need the correct position for them to be turned on and turned off. So delving a little bit deeper into inversions, let's look at what a pericentric inversion is, and this is an inversion that causes a loop and includes the centromeres. Okay, so pericentric means includes the centromeres. So we can see here are 2 chromosomes. Even though the genes are trying to line up correctly, if we get an inversion, then they don't have the ability to line up correctly. So they're going to twist into an inversion loop to try to get those genes to match up during prophase 1. And during that, and within that inversion loop, we're also going to have crossing over taking place, and that is going to result in a dicentric bridge, not a dicentric bridge, but that's going to result in the 2 long arms of the chromosomes actually trying to pair with the opposite chromosome. So here we've got our 1 chromosome, here is another, and we've got the Q portion of both chromosomes trying to pair up with the opposite chromosome. So in this case, no dicentric bridges or acentric fragments are produced. We have whole chromosomes, but it's very, very difficult to keep them from pairing up, because when anaphase 1 happens and they start to separate, those recombinant chromosomes are going to have too many copies of some genes and then no copies of other genes. All right, so again, our results with a pericentric inversion is we get 4 gametes. Okay, we get a normal gamete that doesn't have any issues at all. We get 2 recombinant gametes that are not viable because they don't match up. The centromeres are in the wrong position because of that inverted loop, so they're lost and basically can't function. And then we'll get a non-recombinant gamete with a pericentric inversion. So those might be viable, but a lot of times they are not. So inversions have really played a big role in our evolutional history. So through G banding technology that we talked about when we were at the beginning of this chapter, when we were talking about karyotyping, G banding patterns have shown us that several human chromosomes that we have today differ only slightly from certain chromosomes of chimpanzee. And by slightly, I mean only 1 pericentric inversion. So our chromosome number 4 looks like this, the top chromosome up here in the center of the slide. Where our centromere is at is more towards the right side here. So we've got our Q portion of the chromosome and our P portion of the chromosome, long and short, or short and long respectively. And because of a pericentric inversion from our ancestors, the chimpanzee, that actually resulted in us becoming humans and chimpanzees staying chimpanzees. So these type of inversions, as long as they stay viable, really have helped with the evolutionary process. So keep in mind that with inversions, the order of the genes are really what's being altered here. And depending on what type of inversion we're looking at, that can break the gene apart and destroy the gene function. But it also could destroy the gene function because the gene has lost its position if it requires a position effect to be expressed. So, in summary, inversions can be homozygous and will typically synapse and function normally. If they're heterozygous, then they're going to align very weirdly and pair and form inversion loops. And then if crossing over ends up taking place within the inversion loop, essentially bad things happen and we get non-viable gametes. Because of these deletions. So just keep in mind inversions come in two major types, paracentric and pericentric. So make sure you have a really good handle on what those are and some of the effects of those inversions as well.